The screen is yours.
Okay. Thank you very much, Anne-Florence. Thank you to the organizer for inviting me to speak about this subject that I'm working on from several years now. So the idea that I wanted to propose today is linked to is linking, let's say, phylogeny with the sequence space where we can classify proteins with respect to their function. And the point that I wanted to make is very simple, is that sequence spaces for constructive phylogeny and for constructing, classifying, let's say, sequences by function are different. And what I'm going to do is to try to define the second one, which is not obvious and creates some open questions. So as Rémi Monasson did yesterday, I start with the same slides because indeed all what we do depends on the quantity of data that is present now in our databases. And advancement in sequencing technology produced huge amount of sequences. And if we look at the protein sequences, the one that I'm going to use, we see that there are more than 200 millions of these sequences that are present in this piece, in these databases. And if we look at from where they come, we see that they do come from more than 1 million species. So these type of numbers are actually susceptible to grow up enormously in years to come. And there are already projections where they say that in 2025, so tomorrow, we are going to have that genomics is going to be comparable in terms of quantity of data that are going to be generated to astronomy. So it's clear that computational tools or approaches that explain or try to let us classify these sequences in a way or another are going to be absolutely important for us. So what I wanted to do is to say something about this challenge we have. So they want to organize protein sequences in different sequence spaces. And I do think that there are, and that's what I try to say today, that there are these spaces that do explain or let us build up phylogenies and do explain environments or let us classify sequences by environments and finally functions. And I do think that these three spaces are going to be quite soon probably be defined, and they're going to play a very important role to answer to biological questions that today many times are answered just by carrying the information coming from phylogeny. But if we have spaces that do classify rightly sequences by environments or by function, we shall see, I think, phylogeny be used in the right context and be replaced in other occasions by classifications over environments and function. So this is a bit the effort in general that I'm working on, and many people are trying actually to tackle. And what I do today is to try to say something about phylogeny and functional spaces. So the landscapes of mutations that we work on are very different. We might end up to work or to have to work with the protein families that are very conserved or let's say quite conserved or very diverged, as you can see here. And what I'm going to say is that if we use multiple probabilistic models to look at these functions and the different type of evolution that evolutionary pressures that were undergoing these families, then we are going to be well placed for saying something or classifying these sequences eventually functionally. So how to construct probabilistic models? So supposed to have a protein sequence, usually what we do is to look at homologous sequences. We take them from our databases, and what we do is to try to build up profiles, probabilistic profiles. There are different ways to do it, but the idea is that after the alignment, we're going to study, to analyze the type of distribution of amino acids in each single position of the sequence, and we construct these profiles that are sort of representing the type of distributions that are associated to each single position, so on a sequence. So we do it so by this global consensus. Now, if sequences are well conserved, everything is perfect, fine. We are very happy with our profiles because we have that some positions are more conserved typically than others, and they are very likely associated either to positions that play an important role in structures or in functions. So the point comes when these sequences are weakly conserved. In fact, these profiles, as a result, are weak. There is no signal, and so we shall not be able to do much with them. So we wanted to avoid weak profiles obtained by global consensus, and the question is how to do that. So what we shall do is to take these homologous sequences separately, and then we consider them one by one, and look in our large databases, and for each one of these sequences, we can actually take from the databases sequences that are the most close to each one of these homologous sequences. So we will construct alignments that are associated to each one of these sequences and construct different profiles for each one of the original homologous sequences. So these profiles will be actually usually rather conserved, and they will represent somehow some local consensus in the space of all possible homologous sequences. So the idea is, for instance, take the PFAM library of model library. So you have a domain sequence, you have homologous sequences, and instead of constructing a single global consensus model, you will go into the space of sequences, for instance, UniProt. You are going to see where each one of these sequences is located in the space, and then you will look around these sequences for sequences that are close to it, and construct for each one of them a model to build up a model, a huge model library on the protein sequences you are interested in. So just to make clear and give you some sense of the context on which we can work, if we take PFAM, 15,000 domain sequences, if we fix to construct 150 models per domain sequences, we end up to work with model libraries of more than 2 million models. Yet, even if it looks large, yet I think that it's important to see what all these can actually let us gain from this effort. Meaning that we need to keep in mind also that once we build models for a protein family, then these models are there. They can be used forever, and they need not be redone. So to see how we classify sequences, let me first say something about another problem that will explain what these multiple probabilistic models can bring to our understanding of the evolution of the sequences. So I'm going to speak about another problem, which is domain annotation, and you will see that is going to tell us interesting things about these probabilistic models. then I go to my original question, which is classification by function of these protein sequences. So what I wanted to do here, it's something of the kind. So we take a protein sequence and I'm going to annotate it with domains. So domains are smaller sequences that correspond to some structural unit that has usually functional information associated to it, but it doesn't matter for now about function. So their length is in average, let's say 100 amino acids. You might have very different extremes, small, very, very small domain, very large domains, but let's say we can think about sequences of 100, 150 amino acids. So the idea is to annotate large sequences with these domains, and what we can do is to construct probabilistic models for each domain. And instead of using this global consensus model, we can think of using these local models, but the idea is the same. Once you have a probabilistic model, you hit the model against the sequence, and then you see how well this model fits the sequence, hits the sequence. If you have a full sequence, fine, okay? And if you have a read sequence because you work with metagenomic sequences, it's also fine. You can actually try to find some sub part of hit, but let's say the principle is absolutely the same. So the question is what strategy to follow in searching for domains that might be very divergent. And the idea here is that if you want to annotate a genome, let's say that is here, if you have that this genome has proteins inside that are rather conserved, let's say, okay, or diverge, but not very much, typically you are going to find the way to annotate that is, let's say, around in the same clade, okay, by looking at similar sequences in the same clade of the genomes you're looking at, or of the proteins you're looking at. But if by any chance you have to deal with very divergent sequences, then the point is that you have to change the idea, the approach. So if you take these local models somehow that you constructed by starting from sequences around your space of sequences, and that they are spread all around the phylogenetic tree, well, very often it turns out that these type of models that you are using, and they are close to the sequence you want to annotate, might be very far from the clade you are working on. And to show you that this is the case, I'm going to take Plasmodium falciparum, a genome that we looked at, and there it's rather interesting to see that eventually the 50% of the sequences in Plasmodium of the domain annotation in Plasmodium are actually annotated by models that are generated by original sequences in the same clade, but all other models, okay, are all the rest, so the 50% that is missing is actually annotated by best models that are generated with sequences and with model generated outside the clade of Plasmodium falciparum that is alveolata. So this means that looking at models, at these multiple models can be very important for understanding actually our sequences. Let me say one more thing that I believe it's extremely important too, is that if you look at one protein And for instance, this one, doesn't matter what it is, that was not really annotated by PFAM at that time. It was, PFAM recognized only one domain, which indeed was annotated by similarity with sequences in the alveolar clade. Then if you look at this protein, then you find, by looking far away, in fact, different types of domains, new domains that were not annotated before. But the interesting thing is that when you look at the origin of the models used to annotate these two domains, well, you find that these models might belong to different clades. This goes a little bit on the direction of what yesterday Christophe Desimos was actually telling us, by the way. So just to make a link with other things that have been said here. So, and this is extremely interesting because when we look at a protein sequence, then we see that the evolution within a sequence might take different type of steps. And that protein evolution pathways do not seem to follow rules dictated by phylogenetic organization. And this goes absolutely on the direction of the fact that when we do construct phylogeny, we wanted to use conserved sequences and not sequences that are very far apart. So let me go with our original question, which is this classification of sequence for function, based on function. So now here in the previous question, we search for homologous sequences. Here we assume that homologous sequences are given and we want to classify them by function. So we are here at the same point that we lived before. And we had constructed models here and supposed to have n of these models. So we work with multiple models. So what to do with these models? We start with a number of sequences here. These sequences are the homologous sequences that I want to classify by function. And what I do, it's something extremely simple. You will see, okay? So you take these sequences, each of them, and then you take each one of the models and you look at each sequence by each one of the models. So you are going to rewrite a sequence as a vector where each entry of the vector is nothing else than the score of the hit that the model is actually having when it hits, okay, it is matched with the current sequence. So in other way, in another word, what am I doing here is to take a sequence and look at the sequence by the point of view of n different probabilistic models. And so each model is going to speak, okay? So it's going to give us these, what the ensemble of the models is giving us this vector. And for each sequence, we have a vector. So these vectors now, I can put them in a space where each direction of this space is going to be associated to a model. And it turns out that this type of representation space makes, in fact, a step towards the functions. Okay, let's see. So this representation space can be, if you like, represented once you cluster these points represented by, for simplicity, by a tree, which I call classification tree. The same way you build up in the usual sequence space using distances, for instance, a phylogenetic tree. And what happens is that if I start coloring this phylogenetic tree out of the colors that I found by this classification tree, very usually what I observe is that the topology of these trees is not going to be the same at all. And moreover, what happened is that by using these models, what I can extract is extra information from the model about specific models that can be helpful to separate a given subtrees in this classification tree from all other sequences. And I can construct functional motifs out of this. So now I don't have too much time, Anne-Florence. So how much do I have?
Yes, I mean, it's almost there. So, you know, if you can just...
yes, I can tell you some things.
But rather fast, I think it's good.
Okay, I can take 2-3 minutes. Okay. So I wanted to give you one example on the cryptochrome photoliase family. It's a family which is extremely, well, considered rather difficult, okay, to classify because, well, it is a larger family, spreads all over the kingdoms. There is one specific domain which is important, the FAT domain, but so the structure of these sequences almost the same in terms of domains. There is a chromophore for all of them, and there is the same structure, but the function is very different. So here is the phylogenetic tree associated to the family. And by doing this analysis, as I mentioned, we can construct a functional tree that looks like the one that I mentioned before. And so I go fast. And what we can see is that one important thing is happening. This is the topology of this classification, functional classification tree, in contrast of these cartoons that are representing the topology of the phylogenetic tree. And what we can see very, very clearly is that this topology separates light-dependent from light-independent sequences, and photoliase from photoreceptors sequences in a very sharp manner, which was not possible to realize before out of phylogeny, distant functions. And there is even a subset of these sequences that is not associated to function, and we classified it towards as a photoreceptor. There is only one single sequence that is known as a photoreceptor, but as you see here, it was classified as a photoliase or within a mixture of subtrees that could not distinguish, in any case, its position. So there is a lot to extract out of these analyses, but since the time is not there, so I would just cut it. And one last thing is that, well, all this work has been done by comparing with different tools and different approaches. One can be Panther, for instance. Other is a very, very recent one for classification of spaces like COOP. And then there was as well some work done by Rémy Monasson group with Toubiana that I've seen before here. And well, it works well, and very soon you are going to have, if for those of you eventually that are interested, you are going to have a new preprint on bioRxiv, I guess. So I wanted to thank just the people that work with me. So the two postdocs, the two doctoral students, and Riccardo, which is a postdoc that was with me until recently, and that really worked a lot on the development of this last part. And the biologists that will do the experiments, actually, and that's it. Thank you.
Okay, so thank you very much, Alessandra, for this great talk. And so now we welcome questions from the audience. You can either type your question in the chat or you can speak up. So, you know, if anyone wants to speak up, feel free to do so. Yes, I see Eric. Yes.
Yeah, hi, Alessandra. I was wondering, so when you start sort of taking every sequence and then select a set of sequences that are in its neighborhood and then build a model of those. So I would think that in those models, you have especially many sort of like phylogenetic sampling biases, right? Because it just depends on what you happen to have more sequences from that are very close phylogenetically. They're gonna mess with your frequencies of the letter. So do you have some way of, or do you guys try to correct for this? So what do you do with this issue?
Yes. So it's an extremely important question, okay? Why is this? Because the tendency that we have always is to answer in terms of the efficiency of the tools that we are developing, computational algorithms, okay? But here, I think it's very, very important to construct models by spreading as much as possible, going in the direction of what you are mentioning, spreading as much as possible towards the whole space of sequences we have. The sequences we are having now are many because they are coming also from metagenomics, and we included them. So we try to construct these models not just by using sequences that we classified already as homologous. We just need sequences that are close enough, okay? It's clear that if you start from a set of homologous sequences that you don't know about and it's very very close in space, you don't have this spread, you might have difficulties. But okay, so what you can do, okay, is to try to look at sequences that are not already classified as homologs for a protein family, and try to look for new sequences, which you can do now with these models.
Okay, Martin.
I have a question. Yes. Concerning the, you said that in many cases you annotate sequences by the models which are derived from very distant, phylogenetically distant clades and not so much by the profiles of closer by sequences. Do you have any intuition on this? Because it sounds a bit counterintuitive.
Yes, I know that it's counterintuitive, but what happened is that, let me go back to something that I skipped, which I believe it's extremely important. When we do annotate with a model, what happens is that the information that you look at, it's an information that is encoded in the conserved motif represented by the model. So if you look at sequences, even that are very close or very far apart, that are, I don't know, like for instance, this one that are that belong to different classes, functional classes, you see. Then you look at those sequences with the only with the positions that are conserved within the models. Okay, forgetting all the rest, all other information. So there, okay, you are actually looking at specific information that lets you put together those sequences that are most close to each other functionally. So for instance, let me say, if I can take 2 minutes, I don't know if I have time. So let me take these 3 sequences, okay? They belong to 3 different functions. And these 2, moreover, okay, are actually closer in function. I can tell you this, believe me, okay? But when you look at the sequences themselves, these 2 colors one, these two sequences, so this one and this one, okay, are closer in sequence similarity than the other two. Okay, so phylogeny is actually telling you that these two are closer in the phylogenetic tree. Here, what happened is that if you look at the motif that you find with your model, and only at the positions that are highlighted by the motif, then you will find the differences and you will find the way to classify correctly the two sequences. But you have to restrict the information to what counts. All the background, you take it away, and you do it with the motifs that are extracted from these models. I don't know if this is enough. We can.
Okay, I see the idea. Yes,
you see the idea. Yes, and this is crucial. And well, there are several examples that are opening up, in fact, what's going on, but in a kind of very concrete manner.
Okay, so I think in the interest of time, we should move this very lively discussion to the break in the gather space.